The framing of public health risks often shifts with the discovery of new transmission pathways, and recent developments regarding the hantavirus genus have brought the Andes virus strain into sharp focus for global health authorities. While hantaviruses have long been categorized as zoonotic diseases, meaning they jump from animals to humans, the specific confirmation of person-to-person transmission via saliva marks a significant evolution in how epidemiologists assess the risk to families and partners. Historically, the medical community viewed hantaviruses, such as the sin nombre virus prevalent in North America, as a threat primarily confined to those in direct contact with rodent excreta. However, the Andes virus, ANDV, native to South America, has uniquely demonstrated the ability to spread between humans through close contact. This shift in understanding is not merely academic, it has practical implications for domestic settings and travel, particularly as international health agencies respond to a cluster of cases identified in early May 2026. This virus belongs to the orthohantavirus genus and is known to cause hantavirus pulmonosa syndrome, or HPS, a severe respiratory disease that can rapidly progress to respiratory failure and cardiogenic shock. The recent focus on saliva as a vector of transmission stems from evidence that the virus may be present in the respiratory secretions and oral fluids of infected individuals during the prodromal phase, the early period of illness before the most severe symptoms manifest. For the general public, this means that the traditional advice of avoiding rodent-infested areas must now be supplemented with an understanding of how the virus can navigate human social networks. While the risk to the broader United States population is currently assessed as extremely low by the Centers for Disease Control and Prevention, the confirmed mechanism of interhuman spread underscores why recent outbreaks in confined environments, such as transport vessels, are being monitored with such intensity. The primary concern is that unlike other hantaviruses that require the inhalation of aerosolized rodent urine or droppings, the Andes strain can exploit common human behaviors, including sharing utensils, close conversation, and physical intimacy. This distinction is critical for public health reporting, as it moves the hantavirus from a purely environmental hazard to one that requires active surveillance within families and among travelers. Understanding the background of these viruses is essential. They are carried by specific rodent reservoirs, such as the long-tailed pygmy rice rat in South America or the deer mouse in North America. These rodents do not show signs of illness themselves, but shed the virus throughout their lives. When a human inhales these particles, the virus targets the endothelial cells, the lining of the blood vessels, particularly in the lungs, leading to a massive leak of fluid into the alveolar spaces. The resulting HPS has a high case fatality rate, often cited between 35 and 40% in the Americas. As of May 2026, the global health community is recalibrating its guidance to account for these close contact risks, ensuring that clinical and laboratory protocols are equipped to handle a virus that no longer stays strictly within the boundaries of its rodent hosts. This report will detail the specific developments of the ongoing May 2026 outbreak, the laboratory findings that confirm the saliva transmission route, and the practical steps recommended by officials to mitigate a risk that, while geographically concentrated, carries significant consequences for those affected. The core of the current international response centers on an outbreak identified on May 2, 2026, involving the expedition cruise ship MV Hondius, which had been operating in the South Atlantic. According to reports from the World Health Organization and the European Center for Disease Prevention and Control, a cluster of passengers developed severe respiratory symptoms, prompting an immediate investigation. As of May 8, the WHO has confirmed a total of 8 cases, including 6 laboratory-confirmed and 2 probable infections. Tragically, 3 of these cases have resulted in death, resulting in a case fatality ratio of 38% for this specific event. Laboratory analysis conducted via polymerase chain reaction, or PCR, and genetic sequencing confirmed the pathogen as the Andes virus. This identification is pivotal because it distinguishes the current threat from the more common sin nombre strain found in the American Southwest, which has no documented history of human-to-human spread. The ECDC notified the Netherlands via the early warning and response system after the vessel, operating under a Dutch flag, reported the cluster. Genetic data suggests that some passengers may have been exposed to the virus while visiting endemic regions in Argentina before embarking on the ship. Once on board, the confined nature of the cabins and shared dining areas likely facilitated the subsequent person-to-person transmission through close and prolonged contact. Medical epidemiologists emphasize that while most hantavirus cases globally, roughly 2,000 per year, are acquired directly from rodents, the Andes strain is the only one currently known to spread between humans. This transmission is thought to occur primarily during the prodromal phase, which typically lasts between 3 and 5 days. During this window, an infected person may experience fever, chills, and muscle aches, but might not yet realize they are carrying a life-threatening virus. Experts from the University of Florida's Emerging Pathogens Institute and the London School of Hygiene and Tropical Medicine have noted that the virus can be found in saliva and respiratory secretions, making activities like kissing or sharing food high-risk behaviors in an outbreak setting. The CDC has responded by notifying state health departments in the regions where returning passengers reside, initiating a rigorous contact tracing effort to prevent further spread. In the laboratory, the diagnostic process is complex. Because hantaviruses are often present in low concentrations in the blood, specialized testing is required to detect the viral RNA or specific antibodies. Current surveillance data from 2025 indicates that the Americas saw 229 cases across 8 countries, with a case fatality rate of 25.7%, highlighting that the current 38% rate in the cruise ship cluster is particularly aggressive. Health officials are also contrasting these findings with historical data, such as the 2025 case involving the spouse of a high-profile individual in the United States, which initially raised public concern regarding domestic risk. However, it is important to clarify that official statements continue to maintain that the risk to the general American public remains extremely low, as the rodent reservoirs for the Andes strain are not native to the U.S. The primary focus for agencies like the CDC and WHO remains the containment of the current cluster and the monitoring of travelers who may have had close contact with the infected, defined as direct physical contact or spending prolonged time in enclosed spaces with a symptomatic individual. Prevention and public guidance for hantaviruses require a two-tiered approach, addressing both environmental rodent exposure and the specific risks associated with the Andes strain. For the majority of hantavirus variants, the primary advice remains the elimination of rodent habitats in and around human dwellings. This includes sealing entry points, using professional-grade traps, and ensuring that food sources are inaccessible to mice and rats. However, when dealing with the Andes virus and the newly confirmed saliva transmission route, public health officials have issued more specific directives. The CDC and WHO recommend that individuals in or returning from endemic areas, or those who have been in contact with suspected cases, avoid sharing eating utensils, drinks, or cigarettes. Furthermore, maintaining a physical distance and avoiding intimate contact, such as kissing, with anyone exhibiting flu-like symptoms is strongly advised until a diagnosis can be ruled out. The symptoms of hantavirus pulmonary syndrome typically appear between 1 and 8 weeks after exposure. Early signs are often mistaken for the common flu, characterized by fatigue, fever, and muscle aches, particularly in the thighs, hips, and back. About half of patients also experience headaches, dizziness, and gastrointestinal issues like nausea, vomiting, or abdominal pain. The critical turning point occurs 4 to 10 days after the initial symptoms, as the late phase begins. This phase is marked by the sudden onset of shortness of breath and a cough as the lungs fill with fluid. Health officials stress that there is no specific antiviral treatment or vaccine for hantavirus infections currently available. Consequently, the limits of medical intervention are clear. Survival depends on early detection and immediate referral to a facility capable of providing intensive supportive care. This often involves intubation and mechanical ventilation to assist with breathing, while the body attempts to fight the infection. In some advanced cases, extracorporeal membrane oxygenation, or ECMO, may be utilized to oxygenate the blood when the lungs and heart are failing. Surveillance efforts are being ramped up globally, with national and international health regulations, focal points coordinating across borders to track the movement of potentially exposed individuals. While the 2026 Atlantic cruise ship outbreak has highlighted the unique risks of the Andes strain, scientists emphasize that this does not signify the start of a new global pandemic, as the virus does not spread as easily as respiratory pathogens like influenza or SARS-CoV-2. The transmission requires really close contact rather than casual interaction. As the situation evolves, health agencies continue to study the duration of viral shedding in human saliva to better refine quarantine and isolation periods. For now, the public is urged to remain calm but vigilant about early symptoms if they have a history of travel to South America or contact with known cases. This remains an area of active study, and honesty regarding the limits of our current pharmaceutical options is a cornerstone of the ongoing public health response. For more updates on emerging infectious diseases and global health security, consider subscribing to this channel. The clinical and laboratory investigation into the transmission of the Andes virus has fundamentally shifted following the confirmation of viral shedding in human saliva, a development that complicates standard hantavirus containment strategies. While the medical community has long understood that hantaviruses typically enter the human population through the inhalation of aerosolized rodent excreta, the specific verification of person-to-person spread via oral secretions marks a critical turning point for global health agencies. This finding necessitates a more rigorous approach to surveillance, particularly regarding the monitoring of household clusters and intimate partners. In high-containment laboratories, researchers have identified that the Andes orthohantavirus, primarily found in South America, maintains a unique viral architecture that allows it to persist in the human respiratory tract and oral mucosa during the early stages of infection. Unlike the sin nombre virus common in the American Southwest, which lacks a documented history of human-to-human transmission, the Andes strain is now being treated by agencies like the Centers for Disease Control and Prevention as a pathogen that requires both droplet and contact precautions in a clinical setting. The logistical response to this threat involves a complex coordination between field epidemiologists and diagnostic labs. When a suspected case is identified, clinicians must now prioritize the mapping of close contact networks, which includes anyone who may have shared saliva through the sharing of utensils, drinks, or physical intimacy. Diagnostic protocols have been updated to include the use of reverse transcription polymerase chain reaction, or RT-PCR, to detect viral RNA in not only blood and tissue samples but also in oropharyngeal swabs. This allows for earlier detection during the prodromal phase, the 3 to 5 day window before the onset of severe hantavirus pulmonary syndrome symptoms, such as pulmonary edema and cardiogenic shock. Because the virus is classified as a risk group 4 pathogen, live virus work must be conducted in biosafety level 4 facilities, which are limited in number globally. This creates a significant bottleneck in rapid testing, often requiring local hospitals to ship samples to national reference laboratories, which can delay the implementation of life-saving supportive care. Public health officials are also examining the implications for international travel and shipping. While hantaviruses are not typically associated with the rapid global spread seen in respiratory viruses like influenza, the ability for the Andes strain to move between humans means that a single infected traveler could potentially seed a cluster in a non-endemic region. Surveillance at ports of entry is focused on identifying travelers from endemic areas who present with high fever and muscles, though the long incubation period, ranging from 1 to 8 weeks, makes screening at borders exceptionally difficult. In the United States, the focus remains on rural and semi-rural environments, where the deer mouse and other rodent reservoirs are prevalent. But the saliva proof mentioned in recent reports has led to a heightened state of readiness among state health departments. They are now tasked with educating the public that while the risk of contracting hantavirus from a rodent remains the primary concern, the risk of secondary transmission within a household can no longer be ignored. In hospital settings, the management of these patients has become more resource-intensive. Medical staff are advised to use high-level personal protective equipment when performing procedures that could generate aerosols or involve contact with oral fluids. There is currently no FDA-approved antiviral treatment for hantavirus, though some clinical trials have explored the use of ribavirin with mixed results. Consequently, the core of medical intervention remains aggressive supportive therapy, often involving extracorporeal membrane oxygenation, or ECMO, to bypass the fluid-filled lungs and oxygenate the blood directly. The uncertainty surrounding the exact duration of viral shedding in saliva remains a key gap in the current scientific literature, leading officials to recommend strict isolation for confirmed patients until they are well into the convalescent phase. This data-driven approach aims to balance the need for public safety with the logistical realities of managing a rare but highly lethal disease. The confirmation of saliva as a viable transmission vector for the Andes strain of hantavirus necessitates a dual-track approach to public prevention that balances traditional environmental controls with new person-to-person safeguards. For the general public, the primary defense remains the rigorous management of rodent populations in and around residential and workspaces. Public health officials continue to stress that hantaviruses are most commonly contracted through the inhalation of aerosolized particles from the urine, droppings, and saliva of infected rodents. To mitigate this risk, the standard protocol involves sealing entry points larger than a quarter-inch to prevent rodent ingress, using professional-grade snap traps rather than poison to avoid having rodents die in inaccessible wall voids, maintaining clean food storage areas. However, the procedure for cleaning existing infestations has become more specific in light of high fatality rates. Experts advise against sweeping or vacuuming dry droppings, as these actions kick viral particles into the air. Instead, the recommended procedure involves wearing rubber or plastic gloves and thoroughly soaking the affected area with a disinfectant, specifically a mixture of 1.5 cups of household bleach per gallon of water, for at least 5 minutes before wiping the area with paper towels. This method ensures the virus is neutralized before it can become airborne. The second track of prevention addresses the unique risk of human-to-human transmission identified in recent clusters. Because the virus can be present in human saliva during the early symptomatic phase of the illness, public health guidance for those in endemic South American regions or travelers returning from these areas has been updated. Practical takeaways include avoiding the sharing of personal items such as drinking glasses, eating utensils, and cigarettes with anyone exhibiting flu-like symptoms. Within a household or intimate partnership, if one individual begins to show signs of the prodromal phase, fever, significant muscle aches, or intense fatigue, officials recommend immediate medical consultation and the temporary cessation of close physical contact until a diagnosis is clarified. For healthcare workers, the discovery of saliva-borne transmission reinforces the necessity of stringent infection control measures, including the use of N95 respirators, eye protection, and gloves when treating patients with unexplained acute respiratory distress syndrome. The limits of current medical knowledge, however, remain a significant hurdle for global health security. One of the most challenging aspects of hantavirus surveillance is the long and variable incubation period, which can range from 1 week to as long as 8 weeks post-exposure. This delay makes it exceptionally difficult for epidemiologists to trace the exact moment of infection or to identify all potential contacts in a person-to-person transmission chain. Furthermore, there is currently no specific vaccine or post-exposure prophylactic treatment approved for general use. While researchers are investigating DNA vaccines and the use of monoclonal antibodies, these remain in the experimental or clinical trial stages. The medical community must also contend with the difficulty of early diagnosis. Because the initial symptoms are indistinguishable from common respiratory infections, patients often do not seek specialized care until the leakage phase begins, at which point the virus has already caused significant damage to the pulmonary capillaries. Ethically, the reporting of these developments requires a careful balance between transparency and the prevention of unnecessary alarm. While the phrase extreme risk may appear in headlines, the data indicates that hantavirus remains a rare disease, and the person-to-person transmission is a specific characteristic of the Andes strain, rather than a universal trait of all hantaviruses. The public is advised to remain vigilant but calm, focusing on the fact that these viruses do not persist long in the environment once exposed to sunlight or household disinfectants. The focus of surveillance in 2026 is moving toward better rapid diagnostic kits that can be used in field clinics, potentially shortening the time between the first fever and the initiation of intensive supportive care.